In continuation to my previous video, in the process of glycolysis, where 6 carbon molecule of glucose is converted to 3 carbon molecules of pyruvate in 10-step reaction, where we have seen that the first 5 steps are the preparatory phase and the next 5 steps are the payoff phase. Further, the pyruvate is converted into acetyl CoA, which is 2 carbon molecule in an aerobic condition, and it is done in order to enter the Krebs cycle. Now, this Krebs cycle is the second stage of cellular respiration. Krebs cycle is also known as TCA cycle or the Tricarboxylic acid cycle and citric acid cycle. Now, it is known as the citric acid cycle as the first stable product formed is citric acid, which also has 3 carboxyl groups, and that's why it is also known as tricarboxylic acid cycle. It was discovered by H.A. Krebs, a German-born British biochemist who received Nobel Prize in 1953. In eukaryotic cells, Krebs cycle occurs in the mitochondrial matrix, whereas in prokaryotes like bacteria that lack mitochondria, the Krebs cycle occurs in the cytosol. The Krebs cycle undergoes 8-step process. The first step is a condensation step that begins with oxaloacetate, which is a 4 carbon unit, where it reacts with the acetyl CoA, which is a 2 carbon unit, catalyzed by the enzyme citrate synthase to yield citrate. Now, in the further reaction, the citrate loses 1 water molecules and it forms cis aconitate in the presence of enzyme aconitase. Now, further in the reaction, where the citrate is converted into its isomer isocitrate by an enzyme aconitase, and there is a gain of water molecule. Now, the isocitrate produced, which is a 5 carbon molecule, alpha ketoglutarate, that is catalyzed by isocitrate dehydrogenase. In this reaction, the isocitrate is oxidized and decarboxylated to form alpha ketoglutarate. NAD+ is reduced to NADH and release carboxyl group that form CO2. Also, coenzyme A binds with succinyl group to form succinyl CoA. In the 5th step, substrate level phosphorylation occur. Coenzyme A is displaced by a phosphate group, which is then transferred to GDP to form guanosine triphosphate. In the step 6, which is also an oxidative step, two hydrogens are transferred to FAD to form FADH2. Now the succinate, in the presence of enzyme succinate dehydrogenase, forms the fumarate. In step 7, the addition of water to fumarate catalyzed by the enzyme fumarase forms the malate, which is the last oxidative step, produces another molecule of NADH and regenerates oxaloacetate, which accepts two carbon fragments from the acetyl-CoA for another turn of the cycle. Citric acid cycle produces only 4 ATP per glucose molecules, all by substrate level phosphorylation. At the end of the reaction, it releases carbon dioxide, 2 ATP, 6 NADH and 2 FADH2. Oxidation of NADH and FADH2 occurring during oxidative phosphorylation powers most of the ATP synthesis, which we will be studying in our next videos. If you like our video, make sure you subscribe, and thanks for watching.